Still to come, global warming before the U.S. Supreme Court and understanding the brain. Susan Denser of our health unit has that story. The unit's a partnership with the Robert Wood Johnson Foundation.
Focus, focus. This one or this one? Here, why don't we hold it back here? Okay, good choice.
Marguerite Kirst-Colston works hard with her 6-year-old son, Camden, trying as best as possible to get inside his brain.
There. Down. Okay, pink.
Camden was born with a condition called Noonan syndrome and also with autism. The first explains his short stature and wide-set eyes. The second, his difficulties in communicating and in social interaction.
The middle part of his brain, the corpus callosum, which they think is the cable system of the brain, it's there, but it's small and it's thin. And so Noonan's has a big element of problems with growing. And so they wonder if the Noonan's cause the autism or if it's separate. Go run.
Both conditions are triggered by genetic changes or mutations that affect the brain. The hope that Camden and children like him might one day be spared these diseases is why Kirst-Colston is excited about a major new tool in brain research. The tool is the Allen Brain Atlas, a three-dimensional catalog of all the genes active in the brain. It came about because of this man, Microsoft co-founder and philanthropist Paul Allen.
What we've got is really a general purpose tool for anyone that wants to do any kind of brain research. If they want to look at one gene, if they want to look at a set of genes correlated together, maybe in some disease that might end up having a genetic base. And in the brain, we only understand things, at a small scale and at a very large scale. 95% of how the brain works is unknown.
The Brain Atlas is the first product of a new brain research institute that Allen created in Seattle. He says he spent $100 million of his own funds to launch it, partly because he'd watched aging relatives battle degenerative brain diseases.
I think anybody that's been familiar with a family member that's had Alzheimer's or Parkinson's or Lou Gehrig's disease, these are all very, very tough diseases. And so anything that could be done that would accelerate the development of drugs or other treatments for these diseases would be a fantastic thing.
All diseases, including brain diseases, stem from mutations or other changes in the estimated 21,000 genes that make up human DNA. To help understand these processes better, top scientists assembled by Allen advised him to create a roadmap of genes active in the brain. Alan Jones is scientific director of the Allen Brain Institute.
Genes, when they are turned on, the lingo that we use is we call them, they're expressed. And when they get turned on, they actually take that information and they eventually tell the cell what proteins to make. And so the proteins are the things that go about and do the business of a cell. And what gives a cell its unique character, what makes a bone cell different from a skin cell, from an eye cell, from a brain cell, is what genes are turned on. Of those 21,000, what genes are turned on and at what level those genes are turned on.
It's a great source of satisfaction today to see this vision realized.
Scientist Dave Anderson of Caltech is one of the scientists who advised Allen. He told him that knowing which genes turned on and off in different brain regions would provide unparalleled insights into how the brain worked.
It's like looking at a map of Africa, and in the old days you would just look at the borders between the different countries. Now what you can see instead is all the domains where the different animals live, where the different plants are, where the different human tribes and populations are, and that's what you see, irrespective of the borders between countries.
You can really see the advantage of being able to see this in 3Ds.
The ideal would be to have an atlas of genes expressed throughout the entire human brain. But that would require dissecting a live brain, an ethical and physical impossibility. So scientists at the Allen Institute settled for the next best thing, an atlas of the brains of special laboratory-bred, genetically identical mice. It may be surprising, but 90% of a mouse's genes are identical to a human's. Assembling the atlas required the work of nearly 100 scientists, engineers, mathematicians, and information experts.
So we've got individual microscope slides. Each one of them is barcoded so we can track all of the information. And there, as you can see here, there are very thin slices of mouse brain on each of these slides.
They began by literally slicing apart the brains of several thousand mice. Each slice, just 25 microns thick, or about 1/6 of a human hair, was subjected to special chemical probes to detect the presence of specific genes. Under sophisticated microscopes, the mouse brain cells with specific genes switched on look like this. Special cameras captured thousands of these images.
We've generated over 600 terabytes of raw data, raw picture data. To put that in context, that would fill over 20,000 iPods.
The digital images of the mouse brain cells with genes switched on were assembled into a giant database.
We've assembled a large cluster of computers which simply grind away and process this information and put it into this three-dimensional framework.
The end product is this 3D catalog. It's freely accessible to all on the website allenbrainatlas.org. Users can easily click on a brain section and see which genes are active there.
It is exactly like having a Google for the mouse brain now.
One researcher who's used the Allen Brain Atlas is Dr. Susan Swedo. She oversees autism research at the National Institute of Mental Health.
To be able to go online and just map various areas of the brain and what genes are being expressed in that area is phenomenal. I, in 5 minutes, was able to do what used to take a graduate student 4 years for one tiny little nerve cell connection. And now they have it for the entire brain.
Neurosurgeon Gregory Foltz of the Swedish Medical Center in Seattle hopes his work with the Brain Atlas will lead to new treatments for patients.
Okay, we're all set to go.
Foltz treats glioblastomas, the most malignant form of brain cancer that kills about 17,000 Americans each year.
One of the central questions we have is, how does your tumor compare to the normal brain?
Foltz recently showed the atlas to one of his patients, 64-year-old Dorothy Harper, who was diagnosed with glioblastoma last spring. He told her that her tumor was linked to changes in one particular gene called BEX1. That's a finding he and fellow researchers made with the help of the atlas.
We went to the Allen Brain Atlas to see whether or not this gene was normally turned on or turned off. And what we found, if you take a look at the screen, is that this gene is normally turned on. Now you can see that everywhere that the gene is turned on, there's a little dot. And this is a picture of the normal brain. What do you see there?
I still see a lot of dots on that brain.
There's a lot of dots in that brain. That's because the gene is normally turned on throughout the brain. But in a tumor, in your tumor, it's turned off. So it turns out this gene is very important to keep cells from growing. And when you turn it off, cells grow out of control and can form tumors.
These genomic findings have not yet translated into effective therapies for patients like Harper, but Foltz is confident that one day soon they will.
I'm not sure what to compare it to in the history of medicine, but I can tell you that it's going to be something that we will look back on decades from now and say that we were there when this was discovered. I believe it will have widespread impact on all patients who have neurologic disease.
The Allen Brain Atlas is also providing new insight into the brain's overall complexity. One of its findings is that 80% of all the genes expressed elsewhere in the body are also expressed in the brain. That sheds light on how the human species found new uses for the body's genes as our brains evolved. Another unexpected finding is that most genes active in the brain are turned on in multiple regions, not just one. Anderson of Caltech says that underscores how complex it will be to treat many brain diseases. He cites the example of autism.
More? Okay, go get it. You can get it.
Some scientists think autism may stem from changes in genes expressed in a brain region known as the amygdala. That's a part associated with emotions and social interaction.
Most of these genes that are expressed in the amygdala will be expressed in other regions of the brain. If they show changes in expression in the amygdala, we'll also want to know whether their expression has changed in other regions of the brain, such as the cortex, and that might lead us to clues about the involvement of other brain regions in autism that we didn't necessarily expect.
Swedo of NIMH told us she hopes to use the Allen Atlas, along with tools her institute has developed, to better understand the causes of autism.
I think that we have reason to hope that within our lifetime we're going to know what causes autism, and we're going to have meaningful treatments and prevention strategies. The value of the Brain Atlas is it has just leapfrogged us to the next level of understanding. Okay, ready.
That's gratifying news to parents like Kirst Colston.
If someone like Paul Allen is going to spend his time and money looking into something that affects so many people, then that may have an effect where other people get more interested in it, and we may actually get to treatments and potentially cures or diagnosis, which we just don't have now.
The world will develop new treatments for these diseases over the next few decades, I believe. So if what we did was the key to making any of that happen, that will be just incredibly rewarding for everyone involved.
The Allen Institute's next project will be making a genetic atlas of the human cortex, the part of the brain associated with higher functions, such as thinking.
What are you doing? Come here, little guy.